बता देना बाबा बीच में। भक्ता। Now it's time to start the lecture. Good evening, everyone. Those who have attended the previous sessions must be knowing me. Otherwise, I'll just give a brief introduction for myself. I am Kanika Gupta. I'll be a TA for the course Human Physiology that you guys must be attending on NPTEL. And I'm a PMRF scholar, a PhD scholar from IIT Delhi. Okay. So,
Good
yes.
evening, ma'am.
Good evening, everyone. Good evening.
Good
So,
evening, ma'am.
good evening. Okay, then let's start the lecture. As everyone knows, first we'll do a quick
Good evening,
revision.
ma'am.
Good evening. Good evening to everyone. Okay, so we'll first go for a quick revision of what you guys must have started in the last week, and then we'll take up the questions. Okay, so this is how the session will go. Let's start. Okay, so yes, first of all, I hope you guys must have attended the lectures very carefully and very keenly. Still, if there are some confusions, you can stop me in between and ask, or you can ask me after the completion of explanation. Also, totally up to you. Okay, so I'll start with this. First of all, will you guys must be remembering that in the last lecture also I told you that whatever amount of blood that comes inside the heart must be expelled out also, must be ejaculated also, so that a proper functioning of heart is maintained, and also the amount that is left in the ventricles after a an ejaculation, after a strong systole that should be maintained. Otherwise, what will happen if in every cycle, when the amount of blood is increasing in the left ventricle, when after every cycle, if the amount of blood is increasing, then it will lead to the we'll say collection of the blood in the left ventricle, leading to the disruption in functioning. Okay, so who wants to join? Okay. So the thing which explains this is called as the Frank-Starling mechanism. What this mechanism says is venous return is the flow of the blood that is returning to the heart from the rest of the body. Okay, and the venous return depends on the local requirements in each and every component of the body. For example, if body requires more blood, if it is in some situation like in fight or flight situation, in if the person is doing exercise or something, then in that situation the requirement of the oxygen will be high, so that of the blood. So in that scenario the amount will increase for sure. So this is what they are saying. The heart in turn automatically pumps this incoming blood into the systemic arteries so that it can flow around in the system again. And the ability of heart to adapt to the changing volumes of the inflow blood. If the amount is increasing, then heart will adapt accordingly. If the amount is decreasing, the heart will adapt accordingly. But obviously, in the decreasing situation, if it goes below the resting situation, then we say that there is some kind of a problem in heart. There must be bradycardia, tachycardia, anything that we'll see further in the lecture. So yes, the ability of heart to adapt to the changing volumes of the inflow blood is called as the Frank-Starling mechanism. In other terms, if we'll simplify it, it means that the greater the heart muscle is stretched during filling, the greater the force of contraction and the greater the quantity of blood pumped in aorta. You can understand this in a way like if you are stretching an elastic. If you are stretching it like normally, it will come back normally. If you are stretching it like applying more force, uh, like you were applying the force before, and when you're stretching it applying more force than the before one, then obviously when it will come back to its position, it will obviously exert more force than the previous situation, है ना? So this is what happens here also. The greater the force of contraction and the greater of the quantity of blood pumped in aorta. If the contraction will be higher, so the blood pumped in the aorta will also be higher. So this happens when the incoming blood also increases. In that situation, the outgoing blood also increases, but the amount that is remained inside the heart, that after a a strong systole, it remains constant. It doesn't changes. Okay. So the stretching of the muscle fibers augments muscle contraction by increasing the calcium sensitivity of the myofibrils, because we know calcium is the ion which is involved in the signaling, which signals that muscles that it is time to contract. So obviously there will be a change in the calcium sensitivity on the myofibrils, causing a greater number of cross bridge formation and a greater contraction, a greater force for contraction. Okay, so this is what the Frank-Starling mechanism says. Now, when we say the heart is beating in a rhythmic manner, so there must be some control on the rhythmicity of the heart. This can't, obviously the heart beats own by its own, as it has SA node which generates these action potential, and then the heart undergoes contraction and every function happens. But yes, this rhythmicity maintain the change, because this rhythmicity is ought to change depending upon the situation we are in. If we are in rest, then obviously it will go as it was going. If the demand of the body for oxygen increasing, for any kind of change, then the rhythmicity is also changing with that. So how this change happens, which system controls this rhythmicity, is what we are going to study next. Okay, someone wants to join. Let's see this. Okay, take it. So yes, थोड़ा बड़ा करती हूँ इस picture को. Yes, so I hope you all can see this image. What happens here is that here there are two systems working together. That is one sympathetic system, another is parasympathetic system. Now these both the systems are a part of autonomous nervous system. Okay, so what happens here is uh, one neuron which is arising from the CNS, that is here it is medulla oblongata. First we will be talking about this one. Okay, I'll just on the pointer so you guys can see better. Yes, so first we'll be talking about this system, the preganglionic neuron. This term you must have heard in the lecture, so I am not explaining it here now. This is the preganglionic neuron which will be arising from the medulla oblongata. Then it will go and terminate near heart and form a synapse kind of situation with another neuron which we called as the post ganglionic neuron. Okay, so this neuron which is entering the heart is not the one which was arising from medulla oblongata, but the other one which is called as the postganglionic neuron, and the longer one, this one is preganglionic neuron. This is the parasympathetic way of system. Now in the sympathetic system what happens is the neuron which is arising from the spinal cord, if you can see this is the spinal cord, the neuron which is arising from the spinal cord, it will reach the sympathetic trunk ganglion, and then here it will form a synapse with another neuron which will reach the heart, which will connect to the heart muscles, and this is called as the again postganglionic neuron. And here we can see that sorry, the system here, this in which the spinal cord is involved is called as the sympathetic system, and in which the medulla oblongata is involved is called as the parasympathetic system. Both of these systems have these pre and post ganglionic neurons, and both of these neurons finally supplying either incorporating both of these neurons in the chain, they'll be supplying or directing the heart. Okay, so this is how these things work. Okay, we have a person to join the meeting. No problem. Yes, so both of these systems work in a fashion that the rhythmicity of heart can be maintained. It won't go beyond its capacity, and it shouldn't be less than that also, because if either of the situation happens, it is a problematic situation. It is not a normal situation. Okay, so yes. Now the thing here that you should keep in mind is that the parasympathetic fibers, if you can see here, that the parasympathetic fibers are terminating near SA node and AV node. Okay, parasympathetic supply is till SA node and AV node, and the sympathetic supply is the sympathetic nervous system is involved in whole of the heart. If you can see, these fibers are reaching nearly whole of the heart to most of the portion of the heart. Okay, so you guys must remember this thing is parasympathetic fibers are distributed to SA node and AV nodes, and also the muscles of the atria, because obviously we know SA and AV node are near the atria, and the sympathetic innervation is strong to atria as well as ventricular muscle. Matlab, it will go to atria also and it will go to ventricular muscles also. Okay, so this is something you guys must remember, and you shouldn't get confused in this. Okay, someone wants to join the meeting or not? Okay, the next thing here that we're going to study is, again, as I was saying, I'll just read it out so you guys can get a clear picture. Under normal conditions, the sympathetic nerves discharge continuously so as to keep the minimum rate, the resting stage, we would say. The excitation of heart by sympathetic nerves stimulates the heart rate from 70 to 180 to 200 beats per minute, depending upon the situation. If it is a fight or flight situation, or if the person is exercising or doing some kind of strenuous activity, then it will reach up to 180 to 200 beats per minute. Otherwise, in the resting stage, we all know that 72 beats per minute is the resting condition for a healthy human heart. Okay, also increases the force of contraction, thereby increasing the volume of blood pumped. This is all what sympathetic system does. When there is a situation when the system knows that the body requires more blood, more oxygen, in that situation what it will do, it will increase the force of contraction, thereby increase the volume of the blood pumped, and the cardiac output may go up by 2 or 3 folds. Okay, cardiac output जो increase कर जाएगा, obviously if you can see the number of beats per minute is increasing so much, they are almost getting double, so their cardiac output will also increase. Okay, so sympathetic stimulation increases the rate of the sinus nodal discharge, also the rate of the conduction as well as level of excitability in the heart muscles. Obviously these are all controls, because they have to work on each and every control so that the whole system can work accordingly and the heartbeat rates can increase. So this is what all sympathetic system does. Now, if we can see the distribution, as I was telling you guys, the parasympathetic system reaches only SA node and AV node. This is AV node supply and this is the SA node supply. While the sympathetic system reaches whole of the heart. It supplies to auricles also. It supplies to ventricle also. Okay. Mm, yes. So, now we'll proceed further. We'll see what's next. Now, you guys must remember from the lectures also that in the parasympathetic nervous system and in sympathetic nervous system, as both of them involves preganglionic neurons and the postganglionic neurons, then in preganglionic neurons, the neurotransmitter that is being released from the nerve endings of preganglionic neurons to pass a message to postganglionic neurons is acetylcholine. Okay, in both of parasympathetic and sympathetic, in both of the systems, the neurotransmitter that is being released by the preganglionic neurons is acetylcholine, while the neurotransmitter that is being released by the postganglionic neurons is different in both of the case. In case of parasympathetic nervous system, it is again acetylcholine, while in case of sympathetic nervous system, it is norepinephrine. Okay, so this is something you guys must remember again, because it is important that the neurotransmitter involved in parasympathetic nervous system is acetylcholine, either it is preganglionic neuron or postganglionic neuron, while in case of sympathetic nervous system, in preganglionic neuron, the neurotransmitter involved is acetylcholine, and in the postganglionic neuron it is norepinephrine. Okay, so this is something very important again. Now, when we are talking about the norepinephrine also called as the noradrenaline, okay, which is secreted by which part of the body? The adrenal medulla, adrenal gland, which is present above the kidneys. The adrenal medulla secretes this norepinephrine, or we'll say adrenaline and adrenaline hormones. Okay, neurotransmitters. So, what norepinephrine, when binds to its receptor on the heart muscle cell, when it binds to its receptors, when a message is there in the sympathetic nervous system, so it is a G protein coupled receptor. It is a 7-pass transmembranal protein. What does this mean is there is a protein which passes through the plasma membrane by 7 times. I'll show you the structure further in the slides, that it passes through the membrane 7 times, and so it is called as the 7-pass transmembranal protein, or this is a receptor which is coupled with a G protein. So here if you can see, this is the G protein with which it is coupled. This G protein is further helping in passing on the message inside the cell. So this G protein is coupled with this receptor, so it is called as the G protein coupled receptor. Okay, And one more thing that you should keep in mind here is that this norepinephrine is binding to this receptor, but it is not going inside the cell. It is staying outside the cell. It is just passing on the message inside the cell by binding to its receptor, which will then further signal the G protein, and G protein will further signal the adenylate cyclase. This adenylate cyclase will then further use this ATP molecule, and the cyclic AMP will be formed, and further functioning uh, of the signal will happen inside the cell. So it is very important that you should remember that norepinephrine itself is not going inside the cell. It is just passing on the message through its receptor on the heart muscle cells. Okay, so this is a very important point. Okay, so next thing that we have another people waiting. Okay, जी. ठीक है. So let's see what happens in the signaling. NE initiates a CAMP second messenger. Okay, it is a second messenger system which activates protein kinase. Protein kinase का क्या काम होता है? It phosphorylates the protein, the target protein. जिस जो भी इनका target protein होगा, you should always remember a kinase will always phosphorylate. Okay, it will phosphorylate. It will slow the calcium ion channels, promoting more. It will open the slow calcium ions channel, promoting more entry of calcium ions. And also sarcoplasmic reticulum will release more calcium, as this calcium, as we all remember that in cardiac muscle contraction system we have read that here calcium is activating the calcium channels on the sarcoplasmic reticulum to release more calcium and further contraction, to make the muscle contract. Okay, so myosin increasing, myosin cross bridge cycling. Okay, so what will happen? Calcium will be released from sarcoplasmic reticulum. Then further myosin cross bridging will increase and the contraction will happen. And then after the contraction has happened, calcium uptake pumps will come into action and will remove excess of this calcium from the site of the sarcoplasm. It will move these calcium back into the sarcoplasmic reticulum, and also other pumps present on the plasma membrane will help and remove the calcium from this sarcoplasm and will send it extracellular. Okay, so this is how this norepinephrine works. Now one condition is there is angina pectoris. ठीक है? Angina pectoris क्या होती है? Angina pectoris is a condition in which, sorry, people develop progressive constriction of their coronary arteries. Okay, and a cardiac pain will arise. This cardiac pain will, if you can see, this is hole in this area, the shoulder region, the left arm. So a cardiac pain will arise. Why this pain arises? Because the coronary arteries have some blockage in them. This blockage is called as the plaque. Okay, this plaque will hinder the movement of blood in these arteries, and this hindrance will lead to the pain in the heart. Blood is not getting the sufficient supply, sorry, the heart is not getting the sufficient supply of blood, and when there is no sufficient supply of blood, this condition will lead to the angina pectoris. So what they say, let's read this. So, in most people who develop a progressive constriction of their coronary arteries, cardiac pain, which is called as the angina pectoris, begins to appear whenever the load on the heart becomes too great in relation to the available coronary blood flow. Because the heart is working as it was working, or we would say, like, there is some situation, the person is exercising or something, anything can happen. There is some activity the person is going on, but now the coronary arteries have deposited with the plaques in them. So the blood supply to the heart muscles has lessened, and this leads to the coronary to lessen the coronary blood flow, which will lead to the cardiac pain, and the situation becomes angina pectoris. In this situation is commonly treated with vasodilators. Vasodilators, what will they do? They will dilate the vessels so that the vessels, the area in the vessels increases, and the amount that was supposed to flow in these coronary arteries can flow without any disruption. Okay, so generally vasodilators are used, and the one vasodilator that is mentioned here is nitroglycerin. This nitroglycerin is actually a vasodilator, and it will release the nitric oxide, a gaseous neurotransmitter, which will further help in vasodilation. Okay, you guys must remember this thing, that nitroglycerin is a vasodilator. Okay, so another class of drugs that can be used here is beta blockers. Beta blockers that you must have read in the previous lecture, or sir taught in this lecture only. I just let me just confirm. Beta blockers. Okay, beta blockers is something we had read in the previous lecture. So yes, beta blockers can also work in this situation. Okay, so one thing, sorry. Yeah. Okay, people. So if you remember in the first lecture, in the first lecture only, in the first session, I told you that there are two types of acetylcholine receptors. There are two types of acetylcholine receptors. Those people who attended the lecture must remember that. If they don't, and there are some new people, I'll just repeat the thing again, that there are two types of receptors for acetylcholine, and these two types of receptors are cholinergic receptors and the two types of receptors are nicotinic receptors and muscarinic receptors. Okay, so what are nicotinic receptors? Nicotinic receptors are those receptors which bind to acetylcholine and pass on the signal. These are actually ion channels, but these receptors also have an affinity for nicotine. And where do we get nicotine from? We get it from tobacco plant. So these receptors have an affinity for nicotine also. So that's why these are called as the nicotinic receptors. In muscarinic receptors, these receptors have an affinity for muscarine. ठीक है, muscarine we get from mushroom. So these receptors have an affinity other than acetylcholine. They have an affinity for muscarine also. So these are called as the muscarinic acetylcholine receptors. Okay, so there are two types of receptors. And one more thing that you guys must remember here is that these muscarinic receptors are metabotropic receptors, while these nicotinic receptors are inotropic receptors. Okay, so these muscarinic receptors are metabotropic. That means these are again G protein coupled receptors, while the nicotinic receptors are inotropic. That means when they are activated, they form an ion channel and they let the ions pass through them. Okay, so there is this big difference in the muscarinic receptors and the nicotinic receptors. You guys must remember again. Now, where do these muscarinic or these nicotinic receptors present? So you must remember that in skeletal muscles, we read that nicotinic receptors are there at the neuromuscular junction. Okay, the neuromuscular junction, nicotinic acetylcholine receptors are present. Now here where these and here also in the would say that yes, the preganglionic neuron, when they terminate at postganglionic neuron, there also they have these nicotinic receptors, while in the parasympathetic nervous system, as I told you, it still uses it uses acetylcholine as neurotransmitter in both the situation, the preganglionic neurons and the postganglionic neurons also. So in the postganglionic neurons, when terminate on heart, so the receptor present on the heart muscle cell for acetylcholine in parasympathetic nervous system here is muscarinic type of acetylcholine receptors. And again, as I told you, these are G protein coupled receptors, and these are again 7-pass transmembranal protein. And while these nicotinic receptors are made up of 5 subunits. if you can see it on the in the slides, that these are made up of 5 subunits, and the muscarinic receptors are made up of the 7 pass transmembranous protein, which is again linked to a G protein, and this G protein is further generating the secondary message, which will lead to the functioning of this receptor and the signal. Okay, now here, in specifically in heart muscle cell, I'm again telling you, specifically in heart muscle cell, here the muscarinic receptors, when they are coupled to G protein, they are sending an inhibitory signal. Okay, they are sending an inhibitory signal, not a stimulatory signal. Okay, they'll inhibit, or would say they will send a signal to inhibit the contraction. Okay, not to promote the contraction. Okay, so yes. Okay, as I told you, I'll show you the muscarinic receptors. So these are actually muscarinic receptors. They are passing 7 times through the plasma membrane. If you all can count, 1, 2, 3, 4, 5, 6, 7. It is passing 7 times through plasma membrane, so it is called as the 7 pass transmembranous protein. Now, the muscarinic receptor in heart, these are the receptors. The amino terminal is extracellular and its carboxy terminal is intracellular. That means the amino terminal will remain outside and the carboxy terminal will remain inside. Okay, we have, चलिए, ठीक है. Okay. So yes, now what will we do? The terminals are connected by a polypeptide chain that traverses the plane of the membrane 7 times, as I told you. So these are 7 pass transmembranous protein. Acetylcholine approaches the receptor from the extracellular fluid and binds to the site of receptor protein. Here is the sorry. Here is the site for this acetylcholine in the muscarinic receptor. So it will bind here, and the sorry, कहाँ थे हम लोग? Yes, receptor on the extracellular binds to site of the receptor protein. Now the G protein interacts with the cytoplasmic region of the receptor, and this M2 muscarinic receptor activation is coupled to the opening of, what will they do? When this G protein is activated, it will further, the events will take place and it will lead to the opening of the potassium channels, causing potassium to flow outside the cells, producing a hyperpolarized situation. And we all know hyperpolarized situation means inhibition. The action potential can't travel further, so it will cause a hyperpolarization of the plasma membrane. So inhibition signal will be sent and the heart becomes more refractory to contraction. The rate slows. The contraction rate of the heart slows. So M2 muscarinic receptor cause action in the heart in the following manner. It will work at the SA node and the AV node, and decrease in heart rate and conduction velocity will happen respectively, and the heart atria's contractibility will also decrease. So this is what an muscarinic receptor do, and this happens via what? A parasympathetic nervous system. So you guys should remember this thing. Okay. So next thing is, yes, ECG. Now ECG is, the full form for ECG is electrocardiogram. What we do is we generally study the condition, the rhythms of the heart, whether heart is pumping in the normal rhythm, it is the heart is beating properly, or there is some kind of a problem in some part of the heart. As I told you, it is a four-chambered machinery, it's a four-chambered organ. So if there is some problem in any organ or in any part of contraction, relaxation, whatever, that we get to know from this ECG. Now what different things in ECG denotes is uh, the P wave. Here this P wave denotes when the P wave, this is the ECG for a normal person. If there is some kind of a change in ECG, then the situation is described accordingly. We'll see further. So here this P wave actually it denotes is the atrial depolarization before atrial contraction begins. ठीक है? So this is what, this is actually catching what, the electrical signals. So it will tell us that the atria atrias are depolarizing. The SC node has sent the signal to atrias. They are depolarizing, and obviously we know that after depolarization, what will happen? The atrias will contract. So this P wave actually describes the atria depolarization before the atria contraction begins. Okay. Now this QRS complex, what will it do? This QRS complex will tell us that the depolarization of ventricles has happened. Like the signal has reached to ventricles. Okay, the signal has reached to ventricles and now they will be contracting. Okay, now what will the T wave will signify is, like the in this QRS we saw that the signals has reached the ventricles has contracted. Now in this T wave we'll see that the repolarization of ventricles has happened. Like contraction has happened, the work is done. Now the ventricles must come back to its original state. So this T wave shows that the repolarization of ventricles is happening or has happened. Okay. So this is what an electrocardiogram denotes, and this is what it shows. Again, we can see it from this picture. It will be more clear. So what happens is, as we saw that in the starting of this P wave, if you can see this yellow thing, wait for a moment. Yes, if you can see this yellow thing, then in the starting of this, it shows that SC node has generated the impulse and the atria excitation begins. Means the SC node has generated the impulse, it has sent the signal to atrias. Then, as I told you in the previous slide also that P wave signifies the atrial depolarization. So signal has been generated and the atrial depolarization has happened. If you can see the yellow color, this shows the atrial depolarization has happened and now they will be contracting. Now, what will happen further is this dip for Q and then this RS signal. So, when this dip happens, this means that the signal has reached only till the tip of the ventricles. It hasn't spread it to whole ventricles. It has just reached the tip of these ventricles. Now, when this RS complex, this complex completely forms, that means that the atrias are depolarized now. The signal has reached to whole of, sorry, the ventricles are depolarized now, and the signal has reached to whole of the ventricles, and now they will be contracting. Okay, and then again the contraction happens, and this T wave then further signifies that the repolarization of the ventricles. Okay, so yes, this is what this ECG signifies and shows us. Okay, ठीक है. Okay, so next thing is these are the abnormal ECGs, abnormal rhythms. So tachycardia, if you can see, this is the situation for tachycardia. In tachycardia, if you can see, it means faster heart rate, causes increased body temperature, sympathetic stimulation, or toxic condition of the heart. Okay, anything can be the reason for this tachycardia. So if you can see that it is beating very fast, the interval between the two QRS complex has lessened. Okay. Now the bradycardia is the slow heart rate situation. In, if you can see the interval between the two QRS complexes has increased. That means that the heartbeat is slow. Common in athletes, yeah, vagal stimulation causes bradycardia. Basically, bradycardia is not the normal situation of the heart if it stays for long. Okay, and this is the arrhythmia situation. In this, the rhythms are basically irregular. SN node will maybe it fire, may it won't. So this is an arrhythmic situation. Okay. Now, next thing that we'll be studying is the hemodynamics. The simple term by which we can describe the hemodynamics is, it is the system of pipes. Okay, in hemodynamics, what we will be studying, it will be the system of pipes. Someone wanted to join the meeting. Okay. So yes, what what is the proper definition? Let's study that. The term hemodynamics refers to the principle that govern blood flow in the cardiovascular system. These basic principles of physics are the same as those applied to the movement of fluids in general, because we all know blood is also a fluid. So the concepts of flow, pressure, resistance, and capacitance are applied to blood flow to and from the heart and within the blood vessels. So this whole is what hemodynamics, basically the system of pipes, or we can say the movement of fluid, that is blood in our different pipes like arterioles, arteries, veins, venules, capillaries, etc. Okay, so this is what hemodynamics is. Just a moment. What are they doing? Someone wanted to join the meeting. I can't see it. Admit. Admit. Okay. So, yes, this is what hemodynamics is. This is just a pictorial representation of the amount of blood that will be flowing in a vessel at any point of time. This is just a depiction at the resting stage. This is the amount of blood that flows through a vessel at any point of time, or we would say per minute. Okay. So yes, if you can see that in these the pulmonary veins and the pulmonary artery and the pulmonary vein, the amount of blood that will be flowing at any point of time would be 5 liters per minute. Okay, it would be always 5 liters per minute. Yes, it can increase depending upon the situation, when heart is pumping fast, or there is some kind of situation. Okay, so it differs accordingly. Okay, so the rate of blood flow to each tissue of the body is almost always precisely controlled in relation to the tissue need. Okay, so if a tissue needs more blood at some point of time, they are performing some kind of activity, they are involved in some kind of function, then in that situation, that particular tissue requires more blood compared to the other tissue. So in that situation, what happens is the system is designed in a way that, sorry, the system is designed in a way that the blood required, wherever the blood is required more, it will it will reach there, and in some other part it will be cut it down, so that the amount can be managed, or the requirement can be met. Okay, so what happens is when tissues are active, they need greatly increased supply of nutrients, and therefore much more blood flow than when at rest, occasionally as much as 20 to 30 times the resting level. This this can reach to this much high level. Okay, yet the heart normally cannot increase its cardiac output more than 4 to 7 times greater than resting level. The heart can't beat much faster. It can reach up to 4 to 7 times, but it can't go beyond that, because that is not something its capacity. Okay, so in that situation, what happens is, therefore, it is not possible simply to increase blood flow everywhere in the body when a particular tissue demands increased flow. So, what happens instead, the microvessels of each tissue continuously monitor tissue needs, such as the availability of oxygen and other nutrients, and the accumulation of carbon dioxide and other tissue waste products, and these in turn act directly on the local blood vessels, dilating or constricting them. If they'll constrict these blood vessels, obviously the blood flow will decrease. So yes, they'll dilate or constrict the blood vessels according to the need of this particular tissue or the organ. Okay, so to control local blood flow precisely to that level required for tissue activity. Also, nervous control of circulation from central nervous system provides additional help in controlling this tissue blood flow. So this whole system is designed in a way that every need of the body can be met. Okay, whenever a tissue requires more blood, then the blood supply from some other tissue is lessened down. The blood vessels, the local blood vessels in that tissue will constrict, and this extra. and the blood that is actually retracted from that area will be sent to the tissue which requires it more. Okay, so this is how the system is maintained. Now the relationship among vessels, if you can see that these are the large vessels, the systemic aorta and the pulmonary artery. So these large vessels, sorry, the pulmonary veins. So these are the large vessels which are actually connected to the heart, the vena cavas and all. Now these arteries will, these obviously we know that both these blood vessels, both type of these blood vessels are elastic. They have smooth muscle layers in them, which help them to constrict, dilate or constrict. Okay, so this is very important that you remember that these are the elastic arteries conducting vessels, which will further divide into the muscular arteries, which will be called as the distributing vessels. Then these distributing vessels will further divide and form the arterioles. These arterioles will then give rise to the capillaries. Now one thing that you guys must know, must remember, that in the capillaries also they have some kind of a control for the blood flow, which is called as the sphincters. Okay, these are called as the sphincters. These sphincters they can constrict these capillaries to stop the blood flow, or they can dilate and let the blood flow depending upon the situation or the requirement of the body or the signal they get. Okay, now this is something for you to again remember, that these are the, this is the structure for an artery, this is the structure for a vein. If you can see, the smooth muscle layer here is very thick and the lumen here is smaller compared to the veins. Okay, so the lumen of veins is larger than arteries and the smooth muscle layer is thicker in arteries compared to the veins. Why it is thicker? Because an artery is supposed to constrict to maintain the blood pressure when the blood is flowing through it, so that every part of the body can get the blood. Okay, while in case of veins, it, if we, you guys must remember that one again, that when the blood is flowing from your legs to the heart, the deoxygenated blood, then it is flowing against the gravity. In that situation, there must be some system which will prevent this blood to fall back downstairs. Sorry, downwards in the direction of the gravity. So what will help there is, it is this uh, walls that are present in the veins that will prevent this backflow of blood in the veins, and they are moving against the gravity. Okay, so yes, there is a difference in the structure, in the layers of these blood vessels, and obviously the structures present inside. So the smooth muscle wall in the veins is like that in the walls of arterioles innervated by. Okay, now one more thing that you should remember is that the walls, the blood vessel walls, either it is artery, either it is vein, both of these, both type of these blood vessels are innervated by sympathetic nervous system only. Okay, blood vessels do not have a dual innervation as like the heart. Heart has both parasympathetic and sympathetic, but in case of blood vessels, it is only sympathetic nerve fibers. But in case of artery, if you can see, these are more, okay, more. I would say more signaling is there. Sympathetic nerve fibers are more compared to the veins. Why it is more in arteries? Because in arteries they are supposed to maintain the blood pressure. For that they must get a signal that now you are supposed to constrict, now you are supposed to relax, depending upon the requirements of the body. Okay, so now here the receptors involved are alpha 1 adrenergic receptors that are found on the arterioles of several vascular beds. For example, skin and splanchnic vasculature. When activated, these receptors cause contraction because they are getting a signal from sympathetic nervous system. One more thing here is, these alpha 1 receptors are responsive to both the adrenaline and noradrenaline, both. Generally, in sympathetic nerve fibers, the neurotransmitter released is norepinephrine. But when these sympathetic nerve fibers go to the adrenal medulla, where they signal the secretion of norepinephrine, they simultaneously trigger the formation and secretion of adrenaline also. So these alpha 1 adrenergic receptors are responsive to both the adrenaline and noradrenaline both. Okay. So when activated, these receptors cause contraction. Okay, one more thing here is we are talking about the blood vessels which are related to the cardiac system. Okay, the cardiac system ones. When activated, these receptors cause contraction or constriction of the vascular smooth muscles. The constriction produces a decrease in the diameter of the arteriole, which increases its resistance to blood flow. Obviously, when the size of the lumen will decrease, the resistance to the blood flow will increase and the blood pressure will increase. Okay. This is the structure of a artery. So again, the outermost layer is tunica adventitia. The middle one, the smooth muscle layer, is tunica media, and the inner one is called as the tunica intima, which is lined by endothelial cells. Okay, so in vertebrates, all types of vessels and the chamber of heart are lined on the inside with a single layered epithelium, which is also called as the endothelium. Okay, endothelium. They perform many functions. Some endothelial cells secrete agents into the blood, such as nitric oxide or prostacyclins, that affect the contraction and relaxation of vascular smooth muscles or help control clotting. Okay, endothelial cells also sometimes synthesize active hormones from hormone precursors in the blood or terminate hormone actions by degrading the hormones depending upon their location, the signal, the function, whatever. Okay, endothelial cells also participate in immune responses. They also participate in immune responses. If maybe in some further lectures, I will tell you how they'll participate in immune responses. But they have receptors for immune cells, which will further help in generating the immune responses. Okay. So, okay, the blood pressure at different blood vessels vary differently, as you can see here, as we saw in that picture also, in this picture also, that the blood pressure here, the vena cavas, it is generally sorry, not vena cavas, it is the pulmonary artery. So the blood pressure here is 15 mm of Hg, and in the pulmonary vein it is 5 mm of Hg. And when the blood enters the systemic aorta, the pressure here is 104 mm of Hg, an average, because we know the systolic and diastolic pressure is 120 by 80. Okay. So yes, if you see at different point at a point of time the pressure at different blood vessels, then we can see in case of aorta, the large arteries, the small arteries, and the pressure is quite high, jumping between 120 to 80. Okay, it is somewhere in between that. It is, it is oscillating. While when the blood reaches the arterioles, the pressure is lessened down. It reaches to around 40, like between 60 to 40. And when it reaches to the capillaries, the pressure almost comes down to around 10 15, very low. Like if you can see it here, it is around 20, or you'd say chalo 30 to 15 के बीच में जो है blood pressure comes here in capillaries, and when it reaches the venules it almost goes to very low, 2, 3, 4, 5, something like that. So yes, the blood pressure decreases abruptly in case of the arterioles. It is very high when it is aorta, large arteries or small arteries, but when reaches the arterioles or it capillaries, the pressure drops drastically. Uh, this is also important to maintain the structures, the blood vessels as such. Otherwise, what will happen? The capillary lumen is very small, and when the pressure is very high, the capillary may burst out. So that is why the pressure must be lessened down in capillaries, and this is the sufficient pressure for a capillary. As we all know, capillaries are formed of only a single layer of endotheliums. There is no outer external protective layers on the capillaries. They are only formed of a single layer of endothelium. So they may burst out by higher pressure. So this pressure is what's sufficient for capillaries to function. Okay. So the pulsatile pressure in the aorta that oscillates between 120 mm of Hg and 80 mm of Hg. As the blood flows through the systemic circulation, the pressure falls to 0 mm of Hg by the time it reaches the vena cava, because they don't need pressure. What is vena cava doing? It is collecting blood from every part of the body, every part of the body, and taking it to the right auricle. It doesn't need pressure. It is not sending blood to every part of the body. It is actually collecting blood from every part of the body. So it doesn't need pressure. That is why the pressure in vena cava is very low. It is around 2, 3, 4 mm of Hg or something. So the pressure in capillaries is about 35 mm of Hg, or we could say like 30, 20, 15. It is somewhere that. Okay. So pressure drops drastically in arterioles. Okay. So mean arterial pressure. Generally, when we have a sinusoidal wave, as it is shown here, then we generally calculate mean by adding the two things and then dividing it with two. But here we can't do this because the systole and diastole is of different time period here. है ना? This is not a perfect sinusoidal curve here. The time points for different thing is different. The duration for the different thing is different. So that is why the mean arterial pressure is calculated by this formula. This is the systolic blood pressure plus 2 into diastolic blood pressure, and then it is divided by 3. This gives us the mean arterial pressure for any person. It is considered to be the perfusion pressure seen by the organs in the body. Mean arterial pressure is normally between 65 mm of Hg to 110 mm of Hg. It is believed that mean arterial pressure more than the observed is very serious, because it can lead to the bursting out of your blood vessels. So, mean arterial pressure is greater than 70 mm of Hg is enough to sustain the organs of an average person. If it falls below than this, then it is also a very serious condition. I would say problematic condition. Okay, organ failure may happen because blood is not reaching sufficiently to that particular organ. Okay, resistance to blood flow within a vascular network is determined by these factors. The length and diameter of individual vessel. Okay, the longer the vessel is, it will, uh, you must have heard about the Poiseuille's law, that when the blood is flowing, when the fluid is flowing through any kind of tube, it is flowing in a laminar flow. Then the outermost layer, the layers of that fluid which is touching that vessel, they move very slow compared to the one that is running in between, because they are facing the resistance from the vessel wall. So yes, the length and diameter of the individual vessel is what it offers the resistance to blood flow. Then the organization of the vascular network is also what involved in the resistance to blood flow. Like when the network is very dilated, I would say the lumen size is larger, and then it decreases to small lumen. Then that works fine, because it is in a hierarchical manner, it is decreasing to low, it is dividing into low amounts of blood, and then the pressure is decreasing. Otherwise, if there is some inconsistency in the diameters of the different blood vessels in a series, then again the whole network will be disrupted, the blood pressure will be disrupted, and again the situation is not good. Okay, the physical characteristics of the blood, the viscosity. Viscosity is how much the blood is viscous, how much is the hematocrit of the blood. It also determines the resistance that blood flow feels while traveling through a vascular network. Extra vascular mechanical forcing acting upon the vasculature are also what are involved. Now the effect of hematocrit here is if you can see, the viscosity in water is, if per se it is said to be 1. Let's take it as a base value, it is 1. So the viscosity of plasma is around 1.8 of the blood vessel, while the viscosity in the normal blood is quite higher, and it is 3 times as viscous as water because of the presence of different types of protein cells like RBCs, WBCs, platelets. We have different types of cells in the blood. We have proteins in the blood. We have hormones running in the blood. There are many things that are running in the blood, which makes it 3 times more viscous than water. And this is what a perfect viscosity for the blood. A blood more viscous than this will also be a problematic condition. The heart won't be able to pump properly. It will require more pressure to pump the blood. So yes, the viscosity is one major factor for a heart to function properly. Okay, so Poiseuille's law, as I told you, the cause of this great increase in the conductance when the diameter increases can be explained here, which shows uh, this basically Poiseuille's law says that any fluid which is flowing through a blood vessel is flowing in a laminar flow, and it is they assumed it to be in concentric rings, and these concentric rings, the outermost ring that is near to the vessel, that is aligning to the vessel, will be facing more resistance compared to the one that is in the middle. And the formula for this is this F is equals to pi delta p r to the power 4 divided by 8 eta I. Okay, so F denotes the flow, delta P denotes the difference in the pressure, R denotes for the radius of the tube. So as much as higher the radius, more will be the flow. Yeah. Sorry, the more will be the this velocity of the flow in each ring is different from that in the adjacent ring because of the laminar flow. So the law states that the flow of the fluid is related to the pressure difference. Sorry, guys, I forgot this. So the law states that the flow of the fluid is related to the pressure difference, radius of the tube, and viscosity of the blood, and length of the tube also. So higher the radius and the flow of the fluid, the pressure will be less. We all know higher is the area, the pressure obviously will be less. Okay, so this is what hematocrit means. Hematocrit, the percentage of blood that is cells in, is called as the hematocrit. When we take the blood and we centrifuge it, so the cell volume in it settles down, and the plasma containing proteins and other WBC and all, it comes up in the tube, and the percentage that settles down is what is the hematocrit. If a person has a hematocrit of 40, then 40% of blood volumes is cells and the remainder is plasma. Plasma is proteins. Hematocrit is determined by the centrifuging the blood in a calibrated tube. Average hematocrit value in men is 42% and in women it is 38%. And the viscosity of whole blood at normal hematocrit is about 3 times than the water. Okay, so this is what the hematocrit is. This is Laplace law. This deals with the relation between luminal pressure and the wall tension in hollow structures. It says that when the pressure in the lumen of the tube exceeds that the outside the tube by any given amount, the circumferential tension developed within the walls of the tube is directly proportional to the tube's radius. Basically, it talks about the pressure that blood feels when blood vessel is constricting or dilating. Okay. Next thing is, yes, as I told you earlier also, that when the blood is flowing, uh, the deoxygenated is the deoxygenated blood is flowing towards the heart by the inferior vena cavas, and when it is traveling through your legs, it is going against the gravity. So there must be some system which will help this blood to flow through these blood vessel and reach the right auricle. So what happens is these calf muscles actually help in constricting this blood vessels so that it can develop some kind of pressure and the blood will flow upwards against the gravity. These are the walls that will prevent the backflow of the blood and will help the blood to flow in single direction against the gravity. So calf muscles play a good role in this. And when in older age, when the muscles are that weak or they don't work properly, then that situation, if you can see, there are some veins in which we can see the purplish blood that has been deposited. It is called as the varicose vein situation. Okay, and this is what, as I told you, the sphincters will control the flow of the blood. When the requirement of some other tissue is more compared to the one in which the blood is flowing, then the sphincters will constrict and will not let the blood flow in the capillaries, and will send this extra blood to that tissue which requires it more. So these are called as the sphincters will get signal from very different parts. Okay. Okay, so capillaries, the structure of capillary is also different in different parts depending upon their requirement. Like in liver, they are fenestrated. The the intercellular gap is higher in them, while in case of blood, they form a blood-brain barrier and they communicate with each other through very tight junctions. Okay, so this was what we read in the previous week in the lecture. Now let's come to the questions part. But before coming to that, I would like to ask you people if you guys have any questions. Otherwise, we'll do the questions in the assignment. We'll repeat those questions and then I'll take you to those questions that I have brought for you guys. So, yes, tell me if you guys have any questions.
Excuse me, ma'am.
Yes.
This is not a doubt, but I would prefer if you could put the slides into slideshow since it's not very clear in the screen.
I would surely do that, but actually, people keep on joining, so I had to remove it time and again from the slideshow mode. But I'll put it again if no one's joining, no worries. Any other thing you want to ask? Okay, people. Okay, let's go for the first question. You guys can unmute your mic and answer me. I'll ask these things. Okay, so the thing which is marked as one is, anyone can answer? Answer is written in front of you. You can just answer. If you have any problem, any doubt, you can ask me. Otherwise, please answer, guys. No one's want to answer. No worries, I'll answer. These are arteriovenous anastomosis. Basically, the arterial is the junction of arteries and veins. This is where the arteries and veins combine. Okay. Okay. The thing which is marked as 2 is lymphatic capillaries. This is all the lymphatic vessels. And the one which is marked as 3 is the venous system, and the one which is marked as 4 is the arterial system. I hope this was clear to everyone. No one had any confusion in this. Then we can move to another question. Okay, the next question was this, in which the one which is marked as 1. We revised it today also. What is this? Is there anyone in the meeting? And if anyone wants to answer this question, I would love to hear your answers. Anyone wants to answer this? One is, yes, Kishori, yes. Okay. And two is, one is, okay, I'll repeat after it. This one is tunica intima. The two is tunica adventitia. Yes. The one which is marked as third is
Endothelial cells.
endothelial cells. Perfect. And the one which is marked as fourth is
Tunica media.
Tunica media. Perfect. Very good. Okay. Next question. Which is marked as one is the pressure is somewhat oscillating or somewhat increasing in which organ? Or we'll say which part of this whole cardiac system? This is one is it physical visible to you guys? One is pulmonary arteries. Now the second one is in which the pressure is high, very high.
Aorta.
Aorta, very good. The third is in which the pressure is dropping suddenly, drastically. The third is
Arterioles.
arterioles, very good. And the fourth one in which the pressure has come down to like 30, 30, yeah, 20, 30. These are capillaries.
Capillaries.
Yes, capillaries. Good. Okay. Now again, this question, the one which is marked as one is it is aortic arc. Okay. The one which is marked as two is anyone wants to answer? It is common carotid artery, and the one which is marked as three is right common carotid artery. Okay. Okay, guys. So now we'll proceed with the questions that I have brought for you guys. Ready, everyone? The drill will go the same way that happened in the last session. You guys will write your answers in the comment box and then I'll tell you the right answer. Okay? You guys will write your answers, and I would like if everyone would participate, it will help you to practice and it'll help you to remember things clearly. Okay? So, yes, the first question is Poiseuille's law states that blood flow is directly proportional to. We have four options here, and I would like to know the answers from you people. Write it in the comment box, and I'll tell you if you are right or wrong. I'll see your answers and will tell you. So, yes, Poiseuille's law states that blood flow is directly proportional to. Jaykant, you said B. Okay, any other? कोई और जवाब देना चाहेगा इस चीज का? Okay, Subhashri B, Jayakanta B. It is okay. So the answer to this question is actually C. Vessel's radius raised to the fourth power. If you remember, I told you the formula when we were revising the Poiseuille's formula. I told you the formula that r raised to the power 4 is there in the formula. So the correct answer is C. Okay. Okay. The primary driving force for blood flow in the circulatory system is. We have four options: osmotic pressure, gravity, pressure gradient, viscosity. The primary driving force for blood flow in the circulatory system. Okay, Priyadarshini says A. बाकी लोगों के कहना है भाई? Osmotic pressure. The primary driving force for blood flow in the circulatory system is. Yes, yes, you guys are right. It is the pressure gradient. प्रेशर ग्रेडियंट की वजह से ही तो ब्लड फ्लो करता है ना हमारे ब्लड वेसल्स में और डिफरेंट पार्ट्स ऑफ द बॉडी में पहुंच पाता है. सो इट इज प्रेशर ग्रेडियंट. Okay, the next thing is the largest drop in blood pressure occurs across which part of the circulatory system? The largest drop in blood pressure occurs across which part of the circulatory system? अभी हमने डिस्कस की है ये चीज कि लार्जेस्ट ड्रॉप जो होता है ब्लड प्रेशर में वो कहाँ होता है सर्कुलेटरी सिस्टम में. हाँ जी, कहाँ होता है? It is in. It is in. It is not capillaries. It is arterioles. I told you that pressure falls drastically when the blood is flowing from arteries to arterioles. The pressure drops drastically. So the largest pressure drop across in the circulatory system happens in arterioles. Okay, remember this thing. Yes. Who has raised the hand? Yes, who has raised the hand? Do they have some question or something? Okay, no problem. So I'll move to the next question. Yes. ठीक है. Which law explains the relationship between flow pressure and resistance in a blood vessel? Okay, हम लोगों ने पढ़ा है ये. बताइए मुझे. Which law explains the relationship between flow pressure and resistance in the blood vessel? Answers in the chat box, please. Boyle's law. जयकांत, हमने Boyle's law कब पढ़ा? Okay, बाकी लोग, don't they want to participate in this? It'll be good for you people. So I would request you guys to please participate and please tell the answer for question number 4. Which law explains the relationship between flow, pressure, and resistance in a blood vessel? It is Poiseuille's law. आज ही हम लोगों ने रिवाइज किया है इसको. It is Poiseuille's law. So the answer is B. Okay. So the next question is blood viscosity primarily depends on. अभी रिवाइज किया हम लोगों ने. The blood viscosity primarily depends upon. Yes. Tell me. Blood viscosity primarily depends upon. Kishori says C. Rajatha says C. Kamran says C. Okay, good. बाकी लोग don't want to answer? No worries. So the answer to this question is again what? Hematocrit. Because as we saw, it is a 40%. Hematocrit is what is the normal situation. 40 to 40% is the normal situation. So yes, it depends. The viscosity depends upon the blood cells and basically the hematocrit. Okay. So another question is mean arterial pressure is calculated by which formula? हम लोगों ने अभी पढ़ा था. Mean arterial pressure is calculated by which formula, guys? This is question number 6 I'm talking about. So I would like you to answer me for question number 6. Mean arterial pressure. Okay, the terms have changed here, guys. So it is so it should be mean arterial pressure as we can see it from here. The correct answer will be A for this, and I'll show you. This was the formula. Systolic blood pressure plus 2 into diastolic blood pressure divided by 3. Okay. So this is what the mean arterial pressure is. So the answer to this question is A. Okay. Now we would go for question number 7. Which component of ECG corresponds to ventricular depolarization? We are talking about ventricular depolarization. Come on, people, answer. Answer to this question. Subhashri, you are answering for question number 7, right? Yes, the question number 7 I'm asking about which component of the ECG corresponds to ventricular depolarization. रिवाइज किया था हम लोगों ने अभी ECG में. So it is QRS complex. QRS complex is the complex which shows that the ventricular depolarization has started. Okay, ventricular depolarization is there. Yes, QRS wave it is. Okay, next question is what ion is primarily responsible for a repolarization during the cardiac action potential? Which ion is primarily responsible for a repolarization during the cardiac action potential? बताइए कौन सा आयन है जो repolarization के लिए responsible है? This is question number 8. It is, yes, it is option C. It is potassium ion. Okay, so potassium ion is the ion which is responsible for repolarization. Next is what does the PR interval on an ECG represent? What does a PR interval represent on the ECG? A P wave is there, QRS complex is there. So there's this interval in this between this P wave and the QRS complex. What according to you should it represent? What do you find from the options is the correct thing for this? Tell me. It is question number 9 we are talking about. So it is the time from atrial depolarization to ventricular depolarization because P wave shows atrial depolarization and QRS complex shows ventricular depolarization. So this interval will show the time from atrial depolarization to ventricular depolarization. Okay, remember this thing again. So these are some twisted questions that will be asked from you when you will go for some competitive exam or something. So you should remember these things. Okay. Next question is question number 10. An increase in sympathetic stimulation to the heart causes what? Sympathetic stimulation अगर मिलेगा heart को तो क्या होगा? Will it activate or will it inhibit? What will happen in the sympathetic stimulation? बताइए जी क्या होगा? Jagant, read the question carefully. It is in an increase in sympathetic stimulation to the heart causes what? I'm talking about sympathetic, not parasympathetic. So what will happen in sympathetic? It will increase heart rate and force of contraction. Okay, it will increase heart rate and force of contraction. Okay, next question is what is the approximate duration of the cardiac cycle at rest? क्या होती है भाई एक कार्डिक साइकिल की ड्यूरेशन व्हेन द बॉडी द पर्सन इज एट रेस्ट? Yes, it is 0.8 seconds, right? Okay, next question. What type of blood vessels acts as a blood reservoir? If you guys heard the lectures, the videos from the sir's lectures carefully, you must be knowing this thing. What type of blood vessels act as blood reservoirs? Yes, it is option B. These are veins. Veins act as a blood reservoirs in the human body at any point of time. 70 to 80% of our body's blood is in veins. Okay. Okay. Mm, Next question. What does the T wave in an ECG indicates? T wave क्या indicate करती है कि ECG में? It is. It is what? It is ventricular Repolarization. T wave indicates what? Ventricular repolarization. ठीक है? It is ventricular repolarization. Okay, next question is which of the following increases during sympathetic stimulation of the heart? Again, this same question but with a different language. Now you should answer it correctly. Yes, all of the above. Heart rate will also increase, stroke volume will also increase, and contractility will also increase because sympathetic system works in fight and flight situation. Okay, next question is which hormone causes vasoconstriction and increase blood pressure? Which hormone causes vasoconstriction and increases blood pressure? Which organ is that? I'm talking about question number 15. Question number 15. It is C. Yes, it is C, which is epinephrine. As I told you, in the alpha 1 receptors, both epinephrine and norepinephrine both can bind. Okay, so it is epinephrine. ठीक है जी. Now, if someone has any other doubts, so they can ask me. Otherwise, we'll do these three more questions, and rest 7 8 questions you can practice by yourself. Okay. In which condition does the heart beat too fast? Is it bradycardia? Is it tachycardia? It is fibrillation or heart block? In which condition it will beat too fast? Question number 16. Yes, it is tachycardia condition in which heartbeat will be too fast. Okay. Next question is which type of blood vessel has the highest compliance? बताइए. Question number 17. Which type of blood vessel has the highest compliance? Arteries, veins, arterioles and capillaries. बताइए किसमें होगी? Obviously it is veins because at any point of time, as I told you, 70 to 80% of our blood is in the veins. So they have higher compliance. Okay. Which drug blocks the funny current in cardiac pacemaker cells? बताइए. Which drug blocks the funny current in cardiac pacemaker cells? This is for your knowledge. If you don't know its answer, that's okay, but you should know this answer. So yes, Raja, the answer is correct. It is what? It is ivabradine. ठीक है? It is ivabradine. Okay, so these are some more questions that I've brought for you guys. So you can do it by your own. If you have any problem, you can ask me later. I've also given the answers also. Apart from that, if you have any confusion, you want to ask me something, you can ask. Yes, you can ask me your confusions, your questions. And yes, Sharadindu. Sorry if I'm pronouncing your name wrong, but you want to ask something? You can unmute yourself and ask me. You can unmute yourself and ask me. Whoever has raised their hands, they can ask me what they want to ask. Anyone, any question? Tell me your questions. Any confusion from the lectures, from today's lecture, from questions, anything you want to ask me, you can.
Hello, ma'am.
Hello.
Ma'am, understood also blood pressure increase, ma'am. Epinephrine.
Sorry, can you please repeat?
epinephrine also increase the blood pressure
Yes,
and
yes,
responses, and
yes, yes.
and also aldosterone also function.
This is
Uh,
about norepinephrine and epinephrine. As I told you, when I was showing you guys the nerve supply in blood vessels. Just wait and let me get back to that slide. Mm, here. So they have these blood vessels have these alpha 1 adrenergic receptors. Okay?
yes,
These alpha
yes,
1 adrenergic receptors can be activated by both epinephrine and norepinephrine both, because both
okay,
epinephrine and norepinephrine are secreted from adrenal medulla, and when
yes,
sympathetic
yes, ma'am,
system is signaling the adrenal medulla to release norepinephrine, then it is
yes,
simultaneously signaling the epinephrine to be released also. So these
yes,
alpha
yes,
1 receptors are receptive, actually, sorry, they both, the alpha 1 receptors are actually sensitive to both the epinephrine and norepinephrine. So that is why the epinephrine and norepinephrine both can increase the blood pressure. Okay?
okay, ma'am,
Yeah,
okay, thank you, ma'am.
you're welcome. Anyone, any other question? If you guys don't have any other question, then we can conclude the meeting. If you guys want to ask me, you can ask. Otherwise, we can conclude the meeting. Okay? So you can leave the meeting. I'll conclude this.
Ma'am,
Yes, yes.
when blood is passing through the blood vessel,
Mm-hmm.
in case of artery, in case of vein, and in case of capillaries, what is
Mm-hmm.
the main difference, main significant point in these cases?
Can you please repeat your question? When the blood is flowing through arteries or through capillaries, what is the main
And
difference in the
what
flow?
is the main actual I point out that what is the main difference in case of arteries, in case of vein, and in case of capillaries?
Okay. So in case of arteries, we all know the pressure is very high. क्योंकि उस
Oh,
blood को, when the blood is flowing through arteries, then this blood must be distributed to all the organs in the blood, in the body. So the pressure is very
okay.
high when the blood is flowing through the arteries. While in case of veins, the pressure is not so high because it doesn't needs to be distributed to the body. It is rather being collected from the body and reaching to the right auricle. So the main difference between the blood that is flowing in the arteries or the veins is of the pressure. Another thing is, when the blood is flowing through arteries, it is oxygenated. It is supposed to
Yes,
distribute the oxygen nutrients to the different cells and different tissues of the body, while the blood which is flowing through veins is actually deoxygenated. It is high on carbon dioxide. It doesn't have oxygen that much as it has carbon dioxide. So again, there is this difference between this oxygenated and deoxygenated part. Another thing is, in veins the blood is moving against the gravity also, while in arteries it is moving towards the gravity.
yes.
Okay.
Ma'am, I also question when blood reaches to the my extreme point legs. So how can it go upwards and reaches to the right atrium through the vena cava, joining to the veins. So in there is against the gravity,
Yes, yes, yes.
how
It is
can
against the
how can
gravity.
reach
I'll tell
how
you.
can reach the blood from our leg to heart without against the
Gravity. Yeah, I understood your question. So here, if you can see my slide, these are the calf muscles. Okay,
yeah,
these are the muscles
yes,
that are present in our legs. These are the calf muscles. These are called as the secondary heart because they are performing a function like heart. How they are performing a function like heart is, when there is some movement of leg, the blood is continuously flowing through these blood vessels against the gravity, because this blood is supposed to reach to the right auricle. So these
Yes.
calf muscles when contract, they push these blood vessels also and generate a kind of pressure so that this blood can flow against the gravity. And as I explained earlier also, that veins have walls in them, so these walls will not let this blood backflow towards the gravity and will prevent its backflow. So here the calf muscle and these walls are helping in this, in flowing in making this blood flow against the gravity. Okay.
Okay. Okay.
So veins and calf muscles are the ones that are performing the function.
Yes, there's functioning as just like a heart, like valves is there also.
Yes, yes, yes. Walls are there, so it won't let the blood backflow in the blood vessel, and these calf muscles will further try to create a kind of pressure in the blood vessel so that blood can flow in opposite direction to the gravity.
Okay.
Yes.
Okay. Thank you. Thank you, ma'am.
You're welcome. Anyone? Any other question? Any other questions, people? You can ask me. Otherwise, I'll be concluding the session. Okay. Okay, so everyone can leave the meeting. I'm now concluding the session. Thank you so much for joining. We'll meet next week. If you still have some confusion, some problems, we can discuss in the next meeting also. Okay? So, good night, everyone. You can leave the meeting.
Thank you.
You're welcome. चलिए जी